Mr. Eugenio Rivera, your science teacher, and welcome to my virtual class.
At the end of the last ice age, 10,000 years ago, our hunter-gatherer ancestors began keeping flocks and herds and cultivating crops. Farmers began to selectively breed certain traits into and out of their animals and plants. This has been happening for thousands of years, but the knowledge of the actual genetics behind it was relatively unknown until the last 100 years or so. We have all heard of Charles Darwin, right? What was actually the lesser-known scientist turned monk, Gregor Mendel, that came up with the correct hypothesis for how inheritance of traits work, whereas Darwin, Lamarck, and other leading scientists had failed. Mendel published his ideas in 1866, but it took until the 1900s for his ideas to be recognized. Mendel studied the inheritance of different characteristics in pea plants. He found that when he bred purple-flowered plants with white-flowered plants, the offspring would either be white or purple, not a mixture of the two, or pale purple. He also noticed that this held true for other traits, like the seed and the pod color, either being yellow or green, and not a mix, and the stems being long or short, and not somewhere in the middle. Mendel concluded that inheritance of characteristics is determined by units that are passed from parents to offspring unchanged. You either inherit your mother's genes or your father's genes, and not a mixture. When looking at the green or yellow pea pods, Mendel found that the first generation of offspring from a yellow pod and a green pod parent will have a yellow pea pod, but the second generation will have green pods in a 3-to-1 ratio. This led to Mendel proposing that a trait might not show up in an individual but can still be passed on to the next generation. Mendel had discovered dominant and recessive genes. Watch our video on common terms used in genetics if you're unsure about dominant and recessive genes and genes and alleles. The gene for yellow pods is dominant, and the green pod gene is recessive. Notice how we have the parent generation, and then the first generation is called F1, and the second generation is called F2. Genetic diagrams are great for showing how this works. We have two parents. The yellow pod parent with the two dominant yellow genes, notice how the dominant traits are given a capital letter, and the green pod parent with the two recessive green genes, which are shown by a smaller letter. These are their possible gametes, and these are the possible genotypes of the offspring. Because the yellow gene is dominant, all of the offspring will have the yellow pod phenotype, but do carry the green recessive gene. Now let's see what happens if we cross two of the F1 offspring generation. And in our next generation, F2, we have 3 yellow pods, but this time we have a green pod offspring that has both recessive green genes. Mendel consistently found this 3-to-1 ratio in later generations as well, which led him to his proposal that traits might not show up in an individual but can still be passed on to the next generation. You can be a carrier of a gene that codes for a different phenotype, but not actually show the phenotype yourself. Remember that the phenotype is just what is physically expressed as the green pod color. We can also show this information in a Punnett Square. We will look at Punnett Squares in more detail in our Punnett Square video. So from this video, you should now know that through observing the phenotypes of lots of crosses of pea plants, George Mendel proposed that you either inherit a gene from your mother or from your father, and not a mixture of both. And because of recessive genes, traits may not show up in an individual, but can still be passed on to the next generation, and may show up there instead, as with the green pods.
Mendel proposed that you inherit a gene either from your mother or from your father, but not a mixture of both. In our genetic crosses video, we looked at what happens when yellow pea pod and green pea pod parents are crossed and represented the offspring in genetic crosses. In this video, we are going to look at monohybrid crosses using Punnett squares. A monohybrid cross is the study of the inheritance of one characteristic, such as the pea pod color. A Punnett square is a diagram that is used to predict the outcome of a particular cross. It is used by biologists to determine the probability of an offspring having a particular genotype. The genotype represents the genes of the organism and is shown by two letters. A capital letter represents the dominant form of a gene or allele, and a lowercase letter represents the recessive form of the gene. So the dominant yellow pea pod is shown by capital Y and the recessive green pea pod by little y. Now back to our Punnett squares. We start with an empty grid like this. If we crossed a heterozygous YY and a recessive YY parent together, we enter the first parent's genotype here, split them and enter one Y in the first column and the other Y in the second column. And the second parent here, doing the same thing, one Y in the top row and the second Y in the bottom row. It doesn't matter which parent you put where, both are correct. So these are the parents' genotypes, and these are going to be the possible offspring genotypes. Let's fill in the gaps. I'll do the first one for you. Dominant Y and recessive Y gives YY. Can you complete the rest of the Punnett square? Pause the video, have a think, and click play when you're ready to check. Did you get it right? Make sure you always have two letters in each of the four boxes. We have two YY genotypes, which means 50% of these offspring would have the dominant yellow pea pods. And we have two YY genotypes, so 50% of these offspring would have the recessive green pea pods. We could write this as 1:1 yellow-green phenotypic ratio. Let's look at another example. In the cystic fibrosis video, we've discovered that cystic fibrosis is caused by a mutation on a recessive gene. You can be a carrier but not suffer from the condition. Let's have a look at the probability of offspring being cystic fibrosis sufferers when two carrier parents with the CC genotype mate. Can you complete the Punnett square? Pause the video, work it out, and click play when you're ready. Did you get it right? So there is a 25% chance that the offspring of two carrier parents will be sufferers of cystic fibrosis. We can represent this in a 1:1 ratio. See how easy Punnett squares are for showing probability of offspring genotypes? But remember, these are just probabilities. We call these the unexpected results. So whilst we could expect a 25% of offspring of two cystic fibrosis carriers to suffer from the disease, it is a probability. The actual results may be different. Two carrier parents could potentially have 4 children that are all non-carrier CC genotypes. Before we finish, maybe some of you have been wondering why we are allowed to just split up the parental genotype into 2 separate letters. This is because during meiosis, the cell division that produces sperm and egg cells, the chromosomes are separated into 2 halves. So the dominant C is pulled one way to form one sperm or egg cell, and the recessive C of the other chromosome is pulled the other way to form a different sperm or egg cell. So half the sex cells will have the dominant C and the other half will have the recessive C. So this C represents half of the sex cells, and this C represents the other half of the sex cells. So that is Punnett squares. Very useful for looking at the probability of genotypes of offspring. We will look at codominance and sex determination in different videos.
Alright learners, thank you very much for watching our science lesson today. I hope you learned and enjoyed this session. Don't forget to subscribe and hit the notification bell of my YouTube channel, Survey TV, for you to be updated of my latest and exciting video lesson about science. So see ya next time. God bless.